welcome back to another spaceexplr episode. And we have a very, very special guest on today. So I'm very pleasure to introduce you to Kate Adamala. So she's a biochemist building synthetic cells. Her research aims at understanding chemical principles of biology, creating tools for bioengineering and basic research. So Adamala Lab studies origin and earliest evolution of life, develops tools for using synthetic biology to colonize space, and works on new tools for biotechnology and medicine. She's the one of the leaders of Build a Cell research community, which of course, for more information, there is a website called buildacell.org that you should go and check it out. Again, they have a lot of great stuff, kind of Build a Cell 101, where they're working on workshop, seminar series, and a lot of great stuff, mentoring involved. And of course, Twitter, Kate Adamala, which of course is included in the show notes. So Kate, again, thank you very much for coming over today.
Thank you for having me, and hi.
Thank you. It's a pleasure to have you on. Again, it's a very interesting topic we're going to be talking about. Again, building synthetic cells. That's a lot of people that probably don't hear, on every kind of day-to-day basis. But can you talk about your personal background? I mean, where you're coming from and how did you get involved with Biochemist?
I'm actually a chemist by training. I grew up in Poland and I studied organic chemistry at the University of Warsaw. And then I got interested in origin of life and astrobiology. And it was kind of a discovery for me that you can actually do that for a living. Everyone who grows up watching science fiction, they know there is always an exobiologist on board of every spaceship, and it's this kind of cool thing that's absolutely fictional. And then I discovered that you actually can do that for a living. You actually can be an astrobiologist. So that's what I decided I really want to be. And so I started my PhD with one of the best experts on the origin of life and building life in the world, Professor Pierluigi Luisi at the University of Rome. And I lived in Rome for a while, and then I went first for a short internship to work for Jack Szostak at Harvard University, and then I kind of stayed in Boston. I finished my PhD in Boston with Jack, and then at the end of that PhD, I decided that I would like to, after all, do something useful. Because origins is great, it's a great discipline, but it's very foundational research. There's not many kind of applied practical avenues that you can pursue if you want to just do origins. So I went and did a postdoc in synthetic neurobiology lab with Ed Boyden, and I learned a lot about classical biology and neurobiology. I learned to do tissue culture, animal work, and I decided I don't quite like it. I don't want to be a neurobiologist after all. But kind of in a meantime, I came to the conclusion that there is a way to combine those two kind of interests, the practical applications of synthetic biology with this pursuit of building life, origin of life, figuring out how life started. And that's how I came to work with synthetic cells. It's kind of research area that requires a lot of foundational work, a lot of tool development, but it's also incredibly fascinating. You're basically building living organisms from scratch. So there is a lot of applications that just a few years ago totally sounded like science fiction. And it's also a great place to be for a person like me who's a chemist, because I like to know where my molecules go. I didn't like that about biology, that you study a cell and it's just a black box that you're given and you're supposed to believe stuff is going on there and you don't actually care where every single molecule goes, which was kind of nerve-wracking for me. So working on synthetic cells lets me actually kind of focus on molecules, know where exactly every protein, where every small molecule goes in my cell, and yet this whole system behaves like this most complex phenomena known to us, which is life. So that's why this is so fascinating to me, at least.
Wow. So I mean, that's that's for a lot of people, like, where do we start? Because I mean, there's so much more information, about build a cell, and kind of we're talking about those building blocks to create those living organisms. So first of all, what type of organisms are we talking about, and why is that, first of all, important for us to create those building blocks and create this kind of synthetic, organisms?
The reason why it's important is first because all of the biological research that was done up to now was done on a sample size that equals 1, because we only have one type of modern terrestrial life to study. So all of biology was done on this simple single sample size. You can think of it as this great variety of organisms that we have available, from unicellular little bugs all the way up to the complex organisms like mammals and everything in between. But it's all based on the same architecture. It's all coming from the same last universal common ancestor of all organisms. So we only have one type of life to study right now. And that's one of the biggest motivations behind my work and behind the field of the origin of the building synthetic cells is we can't really say we understand life or we understand biology until we make it, until we actually are capable of engineering it from scratch, and until we have something to compare it with. You can't say you understand something until you know what properties of that thing are unique to what you're studying and what are general phenomena. And we can tell that because right now we only have one type of life, that the one that we represent. So this is the main motivation for me. We have to be able to make life in a lab so we can say what of modern terrestrial life is unique and specific to us versus what's something that's a general property of every living organism possible in our universe. And then another motivation is that life is surprisingly limiting. There is a lot of limitations on living systems that come from evolutionary constraints because organisms evolved to do whatever they need to do to survive, and that's it. So they evolved to do something and then not other things. And we want to do other things. For example, we want bugs to make chemicals that right now are too toxic for living organisms to make. This is really important when you think about the future of human civilization. We are going to run out of oil, and we talk about this as an energy problem, and we say we have solar, we have wind, we have atomic energy, and that's great. And that will replace oil for energy, but we need to find ways to replace oil for petrochemicals. There's basically entire chemical industry on Earth right now runs on raw materials that come from oil. And when we run out of oil, we don't have a source of those chemicals right now. So no more plastics, no more rubbers, no more fabrics, no more computers. All of that is derived from oil. We need to be able to find biological ways of making those chemicals. And those chemicals are toxic. So bugs are smart. Bugs are not going to make those chemicals. They're just going to die if you force them to make those chemicals. So we need to make a cell, make a living organism that is biodegradable, renewable, but can make those chemicals that our industry needs. So this is one of the biggest practical motivations behind the field of building cells. Another motivation, just to me personally, is because it's cool. Because I think we should be able to build life. We should be able to make cells from scratch. And then also for moving beyond Earth, if we want to explore other planets, we need to be able to engineer life forms that help us terraform those planets and help us support human life for long-term space missions. And that includes new medical solutions, new bioengineering, ways to create nutrients and drugs on demand. And for that, we also need to engineer cells that do that. This is kind of a short description of why I think this is the coolest field ever.
And it sounds very cool. So by the way, so what do you think personally? So, because now people are talking about Mars colonization and kind of creating a basis on the moon, and of course, probably going to be looking towards some other, planets in our solar system. So would you think the best kind of option would be to just send those kind of synthetic cells with the probes and let's say kind of different planets and kind of leave those cells to kind of just leave it there by itself, so the evolution will take it over and kind of so we can create something there? Or should go physically ourselves and colonize those planets? What is your personal approach?
I think both. We definitely should go there, but we should definitely go there right now. That's no question about that. But to really live out anywhere other than on Earth right now, as far as we know, we need to change the environment. I don't think we'll ever be able to terraform the moon without building some sort of a a moon-sized sphere to trap the atmosphere, because moon is just too small to contain an atmosphere. But Mars can totally support a breathable atmosphere. The planet is big enough. It cannot create one itself because of lack of plate tectonics. But if you give Mars an atmosphere and then keep renewing it, Mars can be livable. And then to farm Mars, if we go somewhere, we have to start farming. We all, watched the movie where a guy goes to Mars and grows potatoes in Martian dirt. This is not going to work. And the main reason why it's not going to work right now is because Martian dirt, which is called regolith, is full of this compound called perchlorate, which is really toxic to most organisms, most terrestrial organisms. So in order to farm potatoes or anything really on Mars, we need to either remove the perchlorate from the Martian soil, or we need to develop kinds of crops that are okay with it. And so that requires cellular engineering on incredible scale. We basically have to create an organism that can live with or can metabolize a compound that's toxic to all known modern terrestrial biochemistries. So that's another reason why we have to engineer cells, because we have to either fundamentally change the chemistry of the soil on a planet if we want to colonize Mars, or we have to learn how to grow crops that don't exist right now on Earth in that soil. So I think, to answer your question directly, I think we should do both. And I'm getting a lot of criticism for saying that from the planetary protection people. There's this whole movement of people that say Mars is not ours, so we shouldn't terraform it. But I think if we can get there and stick a flag, it will be ours. So I think we should go there.
Yeah, but whose is it?
There is this kind of group that I interact with at NASA that is very serious about planetary protection and keeping.
Let's first of all go on Mars, and then we can discuss the planetary stuff.
Exactly.
I think it's way too early to talk even about that, because now we're getting politics involved, and politics never helps us in a space explorations, because it just takes things way too slow to make it happen. So, but what do you think personally it would be a doable approach? Again, are we talking about kind of changing the molecular cells of potatoes itself would be kind of easier? And again, from, let's say, if we can use that type of example, or just kind of changing the ground of the Mars? Because again, we're talking about the latter, and probably we could do, let's say, some sort of a, let's say acreage or a small space that we can work on, or you think kind of changing the cell of the potato would be kind of easier? And how does that affect a human being? I mean, in the long term. I mean, is there anything else that you kind of working on when it comes to food and that type of thing that would kind of help us to let's say grow in like those Martian environments?
So I think to do it, you have to go really slow. Right now, we don't have the technology to terraform Mars. The field that I'm in is relatively small. It's been about a decade when people have been saying we're actually building cells from scratch, and it's not crazy to do it. So we're not there yet. We don't have the technology that's required to do what we want to do. What we're working on right now is more molecule production in terms of practical applications. So using synthetic cells to make either drugs or other kinds of small molecules, like for metabolic engineering, small molecules that you can use to plug it into some kind of an currently existing chemical process. And the next step, I think, in my opinion, will be to build synthetic cells that can self-replicate. And once they can self-replicate, then we can start thinking of doing, large-scale things like terraforming operations. Or probably before that, what we will do is we will use this technology to clean up Earth. There is a lot of, toxic heavy metal contaminations or oil spills that can be cleaned up with engineered bugs that can metabolize those either heavy metals or oil products.
And how far are we from making these projects happen? I mean, are we talking about, let's say, 20 years in advance, or this is what's happening currently?
This is what's happening currently. I think we're a few years away from having, and by few, I mean single-digit few, years away from having working prototypes like that. This field is moving incredibly fast right now, so this is really an exciting place to be.
Got it, got it. So it's very interesting again from biological level. I mean, because you're talking about, creating a life form, which of course, like, is it gonna be as us? Like, if we're talking about kind of, creating those colonies and sending those cells to, let's say, like interterrestrial dimensions, light years away from us. Are we talking about something that we can duplicate that is going to be same as us, or is going to be different? Like, so what are we going to be talking about?
Probably different, my guess is. Right now, most of what we're working on is based on modern terrestrial life architecture, because that's what we have to work with. But once we have that down, we will start branching out. And I would love to see how we're going to branch out, how we're going to make life that doesn't resemble the life we know, because that's really the goal for me. To build life forms that use alternative chemistries. For example, nucleic acids that have different backbones, or proteins that are made of amino acids that are different than the canonical amino acids. So those kind of architectures will be really interesting to see, to study, and then go a little further. For example, could life work with more or less than 3 types of biopolymers? Like right now we have life that's based on 3 types of biopolymers. We have DNA, RNA, and proteins. Is it possible to do life, for example, without using that middle layer of RNA? Can we just have a genetic polymer and a protein polymer? Or can we just have one type of polymer? Would it be possible to just have life that is based on one type of a biopolymer? Is it possible to have life that's based on some other kind of architecture? Right now, our life is based on cellular architecture. So everything is compartmentalized into those little cells, and every cell has its own genome, and every cell does its own energy production. Is it possible to organize life in a different way? Is it possible to have, let's say, distribution of functions on a cellular level? Like right now, we have multicellular organisms, but they're still made of cells where each cell is for itself. Each cell lives for itself. Can we split it? Can we, let's say, have a specialized population that does energy production and shares that with all other cells in an organism? That's obviously crazy right now because we don't have that technology, but it would be kind of fun to once we get there, can we investigate options like that? Can we investigate things that never happened in terrestrial evolution?
Got it. So how much of that research actually, let's see, because I think a lot of it is happening right now on the planet, right? So how much do you think of that research is being, and development, let's say, when you're talking about kind of splitting cells and creating those other things? So based on, the oxygen level and on gravity, on different factors that we have on the planet?
Right now, we don't do anything that crazy. The whole field right now is mostly focused on getting to the finish line of this giant quest of let's build a living cell from non-living components. Mm-hmm. This is step 1. We need to be able to make cells that are undeniably alive from components that were not alive, and that haven't been done ever before. And this is kind of a tricky goal to have because we don't have a functional definition of life. There's no definition of life that works for every single living organism and never applies to something that's not alive. So we don't have a universal definition of life. So that's kind of a problem for my field because we say we're building life from scratch, but what are we actually building if we don't have a definition of life? So there is a lot of kind of focus in a field of efforts in the field right now that, we want to know what is life by building it. So we want to be able to put the molecules together in a way that will start to, look like life, smells like life, quacks like life. So maybe it's life.
Yeah. So I think there is one thing that is part of this life is, again, because we carry these kind of carcasses and, we as human beings, and you can see actually a dog, because dog is what is just a piece of meat again, and molecules, and that's pretty much what it is. But when you look at the person or a dog or somebody like that, you can sense something, that there is something different besides a piece of meat. You can see that
Yeah.
there's something, goes inside the kind of so-called spirit type of thing. So how do you form that? I mean, how do you form that? Because you can probably create a piece of meat, but how do you form that kind of, like, I don't know how to even describe that, but you probably know what I mean.
Yeah, and that's a very difficult, probably the most difficult part of our research is we can put all the molecules together right now, but it doesn't click. It doesn't
Hmm.
start self-propagating and doing everything that you would expect life to do. So that's why this is so difficult. We need to be able to not just find a way to put together all the molecules in a living cell, but also to jumpstart it, to get everything to work exactly like we're expecting it to work.
Hmm. So, and again, let's say, so what, like in your personal mind, like what is one of the factors that kind of stop you from making that happen? I mean, because of course you're still in the process of searching that. So if you would have an answer, you would, of course, you would give it right now and you will have everything going. But what do you think that could be, again, from your personal perspective that you've currently been working on? What that could be potentially that kind of don't let you to make that kind of artificial life happen so far?
I don't think there is one thing stopping us right now. The biggest problem right now is complexity. We're trying to put together a lot of things, and we don't yet know very well how to control it. Once we figure out how to control this incredible complexity that goes into a living cell, then I predict, we'll be able to have it start doing life, basically.
Hmm.
If you look at the evolutionary history of Earth, it's reasonable to say that life is pretty easy to get started because, as soon as Earth had a crust and an atmosphere, we started having life. So life should be, as a planetary phenomenon, should be easy to get. And so if we put all the elements together correctly in the lab, my guess is it will start being life.
Hmm. And what can you mention, like what elements are we talking about?
We're putting together the genetic system, so all the genes necessary for making a live cell. Then we're putting together all the metabolism, so protein expression system. Right now, we're building our cells not that different from natural terrestrial cells, so it's going to be a central dogma system with DNA, RNA, and proteins. So we're putting all of that together. We're putting energy regeneration systems together where we have to feed the whole thing. So we have to make energy
Hmm.
in the form of ATP and then break down the waste product and remove the waste product. We're also putting together membranes with all the stuff associated with membranes. So transporters, channels, membrane that can maintain a gradient that can be used for generation of energy. So all those kind of key components of the biochemistry of the simplest cell. And most people, when they work on synthetic cells right now, they put together things that look like prokaryotic cells. So there's no internal compartmentalization. There's no organelles.
Yeah. So again, I think some of the stuff we need to maybe simplify at the same time, because there's probably a lot of us who are listening and watching this episode. They're thinking like, what are we talking about? And sometimes I get the same like expression like, what are we talking? Yeah, because I mean, there's yeah, so I mean, what you're working on is phenomenal, because I think it should be duplicatable. And again, of course, there's a lot of research and time and effort that goes into that. But when talking about creating that cell, because we're talking about creating just the cell on itself right now, right? So,
Yes.
and all of that is creating from all the different parts that are synthetic as well. Or are we kind of using the existing, let's say, material, so called, to make that kind of synthetic cell?
A little bit of both. We're using a lot of the machinery that's used to make proteins is natural right now. So we're repurposing what bacteria and what all the other cells are using. But then a lot of the things we're putting together from scratch. For example, this control of which genes get expressed at any given time, that's designed from scratch because the natural systems are too complex to reproduce. Same with membranes. That cell membrane that we use for synthetic cells is much simpler than the natural cell membrane.
Okay. Okay, let's just leave it there. But one thing that I wanted to ask you is, again, what is lipsomal bioreactors?
So liposomes is just a fancy name for a cell membrane. In our field, we call liposomes everything that's made of lipids, so the same lipids that make your cell walls. And those lipids can be made into those little compartments. And so, liposomal bioreactors could just be a fancy way of saying a cell, because every cell is a liposomal bioreactor, because it's a lipid membrane that encapsulates a set of enzymes that does chemistry, and that's what bioreactor is. When we say liposomal bioreactor, we want to differentiate that from an actual cell. So a cell is a cell, and that's a living cell right now. And a liposomal bioreactor is essentially a soap bubble that makes proteins, because that's what a liposome is. It's a membrane that's not unlike a membrane of a soap bubble. If you ever made bubbles out of soapy water by blowing air in them, that's the same process that's similar to the process that we're using to make liposomes for research. And so a liposomal bioreactor is basically a membrane that encapsulates some enzymes. So in that way, we're all liposomal bioreactors.
Got it. So if you just continue to explain us on a blown bubble kind of level, we definitely will follow you right there. But again, one of the things that you mentioned, again, creating those kind of drug development you're being involved with that when it comes to creating those synthetic cells. So can you talk about what drugs are we talking about and where those drugs would be applied? Is it kind of day-to-day basis, or there's something else?
Both. Right now, most of those drugs would be used only for very specific applications. So some of the things we're working on is targeting rare cancers. So basically using synthetic cells that can go into the patient and find a site of a specific tumor that's very hard to treat otherwise. Kind of more out there applications would be making drugs for astronauts, where during long-term space mission, you have to be able to treat diseases that you cannot predict. So you know that you're sending these people out there in a tin can to, let's say, go to Mars and back. That's going to be a few years. They're going to come down with diseases that we don't know in advance about. So we can't predict that in a year, this astronaut will need a treatment for this kind of a metabolic disorder or cancer. So what we have to do is we have to have a way to make drugs on demand in small quantities very fast. Because when an astronaut gets sick, you don't have that much time to treat him most of the time. So we need a basically bioengineering platform. We need a way to make drugs that we don't know in advance what are we going to need to make. But when we do learn what we need, it has to be made within hours. But we need to make a small quantity of it, because it's most of the time will be just for one patient. So this is something where synthetic cells are really good at, because they're so simple. It's really easy to design them to make something really quickly, because there is none of that biochemistry inside a cell that interferes with all of modern life cell metabolic engineering. So, when you want to make a bacteria, let's say, that expresses certain proteins to make a certain drug, that bacteria has no interest in doing it. It's not on your payroll. You're not going to convince that bacteria to do it out of the goodness of their heart. You have to find a way to put the protein you want to make into the existing metabolism of the bacteria. So it basically, it has a whole bunch of fancy names, but what it comes down to is that bacteria has its business, which is to grow and replicate, and you want to convince it to make something else for you. And that's a hard uphill battle, because you have to make that protein so that it fits into that whole metabolism. This is not a problem with synthetic cells, because we made them from scratch. We designed them, so they don't have their own life agenda. They will make whatever I want them to make. And so that's why this platform is ideal for making small amounts of drugs on demand on a short notice, because you don't have to jump through all those hoops of convincing a cell to make the protein you need. You have a sick astronaut, you diagnose him or her, you decide, okay, this is the drug we need. You make a DNA, so you synthesize a DNA for that drug, you put it into a synthetic cell, and the synthetic cell is not going to come up with a question saying, why am I doing this? This is not in my life plan. Because you made the damn thing. You made it from scratch. So that's why it's kind of very good platform for quick metabolic engineering, for making quick changes to the whole metabolism of and producing different kinds of proteins. And the reason it's mostly for small amounts of material is because it's not a very efficient system. Those synthetic cells are not alive, so the yields of the proteins are much lower than you would do in natural cells. But that's okay if you're just making small amounts.
Okay.
If you just need small amounts, like for one patient.
Got it. So, and that's very good. So, I mean, it's very similar to, again, to coding. So whatever you're going to input, you already kind of know what you're going to get. And again, you're in control of that cell. So that's perfectly fine. But let's say, what about creating that type of medicine and knowing, like, first of all, is it being already tested?
Um, it's being tested in animals.
Yeah.
So there is work out of Avi Schroeder's lab at Technion, where they demonstrated that synthetic cells can be used to shrink tumors in live mice. So this is where the technology is right now, is we have a way to use it for in animals, basically in mammals. As far as I know, there have not been a human trial yet.
So any predictions for that happening soon? Because I mean, we have a lot of kind of health issues that I'm sure a lot of people who are watching and listening, if they probably know a close person of theirs who kind of have those similar problems, they would look to get rid of them as soon as they can. So any predictions when they're going to be start using those on people as well?
My guess is within the next 5 to 10 years, we'll see human trials. The difficulty with human trials of those synthetic cell therapies is that it doesn't fit into any of the boxes that regulatory agencies have. Because regulatory agencies expect drugs that are very well defined. Let's say this is my molecule, this is my drug, and the formulation contains 14 ingredients, and I know exactly concentration of every one of them. And then on the other hand, regulatory agencies are capable of approving a therapy that's based on live cells. So these are, let's say, your own white blood cells that were taken out, modified, and put back into you. What we're proposing is something in between. It's a well-defined biochemical system, but it still is a dynamic biochemical system. Synthetic cells are, doing chemistry, doing metabolism. So you cannot at any given time say this is to the, fifth decimal concentrations of every single molecule that's in your sample. So it doesn't fall into the approval pathway for those one or few compound drugs. But it's not a natural living cell, so it doesn't fall into that category either. So this is kind of a gray area where our field is in right now, and we're early in this process, so we're working on also on lobbying regulatory agencies to develop a pipeline for approving those technologies.
Got it. So what do you think is going to take a long time? Because rules and regulations and everything that's has to do with politics always takes a long time. So you think it could be a problem, or would you think people would kind of take it lightly, as another drug that it can use?
I think people won't take it lightly, and that's good, because we should keep the pipeline safe. This obviously might mean it's going to take slower, but we need to do it safely. And so I think what's needed is kind of change in a way FDA and European regulatory agencies and agencies in other countries look at the ways in which you define purity of a drug. I don't think this will need any changes into the efficacy of the clinical trial protocols. Things like side effects, actual effects of the therapy, all of that. This is a drug, it should fit with that pipeline. But the technical challenges with making a drug, like how pure it is, how exactly you need to know the concentration of everything in that drug, that has to be adjusted, because otherwise you won't be able to use anything that's a synthetic cell, because there'll be living cells that have changing biochemistry, so they can't be that well defined.
So if we're talking about those kind of synthetic cells, so let's say if you take a pill, whatever form is going to be, liquid pill, whatever that might be, and you say you have that kind of problem and you're looking to get rid of that. So is that kind of, gonna have a long-term effect on the body? Because everything that requires for us to kind of swallow, and if you do kind of big doses, and especially if you have a a big problem that requires big amounts of doses, and of course that will have a long-term effect. So let's say instead of using, let's say, chemotherapy, or to get rid of these type of pieces with the synthetic cells, is that going to have a long-term effect?
That's to be found out. That's what clinical trials are for. Essentially, it shouldn't, because those synthetic cells that would be used in therapies, at least in the nearest future, would be made of the same kinds of compounds that natural cells are made of. So after it has done its job, it's had the therapeutic effect, it should be metabolized like any other biological compound that you inject into a patient. So there should be no cumulative long-term effects. But obviously that's what clinical trials are for, and that's why clinical trials take years.
Mm-hmm. Got it. So if you remember, ask you a question about making those type of researches again in the zero gravity type of conditions, which I heard something before that those type of medical research is happening on ISS. So do you personally know about anything like that happening on like in zero G, some type of research in that nature?
No synthetic cell biomedical research happening on ISS right now. Space biology aspect of synthetic cell research is relatively new. Just actually 2 years ago, my lab was, as far as I know, the first group to prepare a synthetic cell payload that made it into space. And so we're relatively early in that stage. The next step will be ISS research with synthetic cells, either in vitro or perhaps, if we're lucky, animal research. I think we are going to do human research on Earth first before we start making biomedical research on synthetic cells in space, because obviously astronauts are much more precious than big patient populations on Earth, where the sample size is much smaller and you need to really know something works on Earth before you inject it into an astronaut.
Yeah, exactly. So what about, again, because we're talking about kind of solving problem, those type of issues, which of course problem solving is a lot of times, like we're focusing that probably 80%, because we're looking kind of on a day-to-day basis, just in general, we're looking to solve problems all the time. But what about, again, keeping people alive? Because we're talking about with your personal research and maybe personal experience, have you seen something that we're creating kind of, let's say, from from a drug perspective that kind of help us to support our activities, let's say in a time when we're going to be colonizing Mars or Moon, because again, because of the lesser gravity and because of the conditions, we're talking about probably decreasing bone structure and muscle kind of tissue. Yeah. So is there anything happening there to kind of support us physically?
Absolutely. For that, I don't think you need novel applications like synthetic cells. There are good drugs right now that are being tested both for muscle and bone loss during long-term space missions. And I think those are problems that we know we will have, so we can prepare in advance. Where the advantages of synthetic cells lies more in things that we cannot prepare for. This is kind of more versatile platform to give you drugs on demand that you cannot plan for, drugs that you can't even think about needing right now. Things like radiation sickness, bone loss, muscle loss, these are things we know we will have to deal with. So there's already research in those areas.
Got it, which is good, definitely. So wow. So I mean, a lot of great different stuff to cover here, and we probably need to do probably 3-4 different podcasts just to cover everything that you're going through, because I mean there's a lot. But can you talk about, again, because I've seen on the website that you do some workshops, so can you tell for the people what's involved in those workshops, and are they available right now?
The Build a Cell community, which is the international synthetic cell community that I'm involved with, runs a weekly seminar series where every week on Monday at 4 p.m. Greenwich Mean Time, we meet and we have a talk about some aspects of engineering cells. We also have twice a year, we organize Build a Cell workshops, which are international workshops open to anyone, regardless of whether you are or you are not a member of the Build a Cell. You're always welcome to come. And during those workshops, we actually work. So those workshops are not just a series of talks. They're actual meetings where people just sit down in working groups and get to work. We have several working groups that investigate different aspects of synthetic cell engineering, all the way from actual hardcore research to education, outreach, policy lobbying, and popular outreach. So there's something for everyone.
And the closest one that's going to be is 19th of February is next year?
Yes. And it most likely will be a virtual workshop.
Yeah, I see that. Yeah, okay. Hopefully not, because I mean, you cannot do a lot of physical activities in virtual meeting, but yeah, okay. So we'll see. So again, wow. So I mean, the last question that I have for you is from what you're currently actually working on, and again, your personal journey. You got involved with it, and it took off like a rocket for you. And I mean, you continue to work on building those synthetic cells, which I think it's such an important piece for us to learn how to recreate synthetic cells and duplicate what we already have around us. And let's say if we want to create something that is hundreds of millions of light years away, and we just gonna probe it, and here we go, 20 years later, 10 years later, there's going to be somebody there and we can go and kind of visit them. So I mean, we're talking about kind of sci-fi stuff, but it's not sci-fi because it's already happening, so that is awesome. So can you talk about, again, what's your personal mission that you're looking to accomplish, kind of footprint and a legacy that you're looking to leave?
I would like to actually make a cell. I would like to be able to put all those molecules into the lamest, weakest possible a cell. I would like to be able to take something that's not living, so all the molecules that are dead in my test tube, and put them together. And as a result of my work, this will start being alive. This act of creating life from scratch, except in a scientific way. So basically putting together all the components that are needed for a living cell. And if I'm part of this initiative and part of the group of people that will be able to achieve that, that would be the most fascinating thing ever for me.
Well, it sounds fascinating. We're talking about, that a lot of people would call a God's work,
It is. It is or it isn't. You can also think about it that way. We wouldn't be doing it if a God didn't want us to be doing it, right? There could be a lightning striking my lab right now, and if a God doesn't like what I'm doing, and it didn't happen yet, so maybe we're doing okay.
Yeah, definitely. No, you're on the right track to do the right thing, because this is definitely a right thing to do. Is just like anything else that you guys see, people involved with the space engineering, space technology, astrophysics, and again, synthetic labs, and you being a biochemist is one of those things that we should pay attention to and focus on. It's not about just Elon Musk and, let him take care of this, thing, because there's a lot of different moving pieces when it comes to life. And again, this piece that you're working on with your team is kind of a big piece, and it's very important. So guys, make sure to get in contact with Kate if you have questions, which I'm sure you're going to have plenty after this episode. So follow her on Twitter. Again, for more information, go to buildacell.org, where you can actually register for upcoming workshop. Maybe you can provide a mentorship as well, and just see what their team is look like and what they're going to be working on in the next future, very soon. So Kate, again, personally, I want to say a big thank you for coming over today, sharing your personal journey and all this kind of mind-blowing stuff that we kind of, at least myself, try to understand, today. So I appreciate it.
Thank you very much. It was a pleasure to talk to you.
Thank you. So guys and girls, one thing that I want to ask you, if you send this episode to a friend of yours, could be any friend, doesn't matter, make sure to send this episode and let them know what's happening with those synthetic cells that are people creating, looking to duplicate life, creating those type of pieces. And, just for inspiration and a lot of information, because there's a lot of great things that Kate's been talked about that, of course, will require your own personal research. So Kate, again, thank you for coming over today. And as always, guys, I'll see you next time. And don't forget, keep exploring.